Epitope. Antigen is a substance which is identified or recognized by the host's body, or in our case, human body, as foreign, and, as a result, an immune response is stimulated towards it. Antigens can be foreign or self. Let us see what does it implies. Foreign antigens. Foreign antigens are also known as heteroantigens. As the name indicates, these substances are not part of human body. They originate from outside the body. They can be microbial agents. We are saying agents because these include whole microbes or their parts. For example, capsules, cell walls, flagella, fimbriae, toxins of bacteria, the coats of viruses, or any surface component of microbes. Foreign antigens can also be non-microbial agents. Non-microbial agents may arise from animals, plants, other humans. For example, pollen, egg white, serum proteins and blood cells from another individual, transplanted tissues and organs. Self-antigens. Self-antigens are also known as autoantigens. Although, as stated earlier, our immune system is able to distinguish between self and non-self, but sometimes our body provokes an immune response against its own cells and molecules. Rheumatoid arthritis is an autoimmune disease, in which immune system attacks and damages own tissues. Now that we understand the term antigen, let's move on to the next term, which is epitope. If you remember previous lectures, we said that the immune system does not recognize whole pathogen but portions or parts of these agents. We termed these portions or parts as antigens. Now, if we explore what is the chemical nature of these antigens, we'll find that most of these are large protein molecules, some are polysaccharides, and a few are glycoproteins or nucleoproteins. Our immune system recognizes these antigens by recognizing three-dimensional shapes of regions called epitopes. They are also known as antigenic determinants because they are the actual part of an antigen that determines an immune response. Each antigen can have several epitopes. Let's understand this by taking an example. We know that most antigens are proteins. So when antibodies bind to the antigens, which are protein in chemical composition, antibodies do not bind to the whole protein, but they bind to a segment of that protein which is known as epitope. Now this segment consists of few amino acids. These few amino acids forms the epitopes to which antibodies bind. It means a single protein antigen can actually contain many different epitopes to which antibodies can bind. Epitopes can be defined as regions or sites of an antigen that are recognized by the immune system, and as a result, they bind to specific antibodies or T-cell receptors. So, we now understand that an epitope is a part of an antigen.